The T lymphocytes are the major players of the adaptive immunity. So in this video, we're going to be taking a look at the development of the T lymphocytes. What happens before we actually get a mature T cell? Now, this is the bone marrow. Just like every other blood cell that develops in the bone marrow, the T lymphocyte does not attain full maturity in the bone marrow. So you know that the B cells attain their maturity, develop and attain maturity in the bone marrow. However, in the case of T cells, that is not the case. The T cell has to move to the thymus gland to undergo certain types of training before they can be fit to be released as T cells, mature T cells. So we're going to be discussing what happens in all this journey. But before the T cell goes to the thymus to be trained, something happened. It doesn't just happen by random chance, okay? The thymus does something. The thymus releases some chemokines that jumps into the blood vessels. So the thymus releases some chemokines like the thymosin, the thymopretin, thymic factors, and all others. Now, these chemicals will attract the T cells. Now, at this stage, this is called the precursor T cells. The precursor T cell, because it doesn't actually develop the surface protein that will actually make it a T cell. So, this chemical begin to drag the precursor T cell into the bloodstream. And from the bloodstream, it finds its way to the thymus, where it is going to be trained. Now, when the precursor T cell gets to the thymus, remember the thymus is actually the training ground of every T cell. So the T cells must be able to pass every stage of the training for it to become a mature T cell. So remember, when we talked about the thymic cells, they secreted certain chemicals, and we said they were called thymic factors, right? The thymic factors and the thymotaxin and all of that. Now, what does this chemical do? It begins to work on the genetic material, right? The DNA of this precursor T cell to secrete important products called the RAG1 and RAG2. Now, these chemicals are very important because they are recombinase proteins. Okay, now I'm going to tell you the function of these chemicals. The RAG further acts on the specific parts of the DNA now to produce certain types of molecules on the surface of the precursor T cell. And the first molecule it produces is what we call the T cell receptor, TCR, the T cell receptor. Okay, now why is this chemical, the RAG1 and RAG2, important? It is important because they have the ability to reshuffle the genes. Okay, so they can reshuffle any type of TCR to fit in any type of antigen. So TCR does not come in one shape, it doesn't come in one form. It comes in different form as a result of the RAG, because it can change the makeup of this molecule so that it can fit in any type of antigen that comes. Now, from this stage, the RAG also manipulates the DNA to begin to produce other type of molecules on the surface. And first one it produces is called the CD4. And it also produce another type of molecule over here, and we call that CD8. Now, this particular precursor T cell is CD4 positive, CD8 positive, and T cell receptor positive. Okay, so let's move to the next stage. What happens here? Remember, we already have our T cell receptor, right? As one of the molecule on the surface of the precursor T cell, and we have our CD4 and our CD8. CD8. Now, at this stage, we expect that certain type of binding will occur between the T cell and the thymic cell. And usually, the CD4, the CD8 molecule will always bind to a particular receptor or a particular molecule, which we call MHC class 1. So MHC stands for major histocompatibility complex. So the CD8 will always bind to the MHC class 1. Meanwhile, the CD4 will always bind to the major histocompatibility class 2. So you get the difference now. The CD4 will always bind with MHC class 2. The CD8 will always bind with MHC class 1. Now, this is actually what we are looking for. If it happens like this, then we now know that positive selection has occurred because the cell has been able to recognize the MHC molecules on the thymic cells. You get? So the next thing that happens now after the positive selection is the next stage. So this next stage, we know we already have the CD4, the CD4 molecule, and we have the CD8 molecule. Now it is time to check if the TCR, which is the T cell receptor, will be able to recognize self-peptides. Now, watch closely. On the thymic cells, the MHCs, so this is the MHC class 1, that will always bind with CD8, and the MHC class 2, MHC class 2, that will always bind with CD4. Now, on the surface of this MHC, there's what we call self-peptides. Self-peptides. Now, our major aim at this stage is to check if the T cell receptor, we already know that the CD4 and the MHC are already good to go, right? They've binded successfully. But we are now checking if our T cell receptor will be able to recognize this self-peptide. So if our T cell receptor recognizes the self-peptide, it is not a good thing. It is a bad thing because we do not want the T cell receptor to begin to attack self-antigen, to begin to attack the cells of the body. Because if it passes this stage and goes out of the thymus, it can begin to attack the cells of the body, and that will lead to what we call autoimmunity. So, in order for us not to allow this cell to move to the next stage, this cell will die, and that is what we call programmed cell death. Programmed cell death, or you call it apoptosis. So if the TCR recognizes the self-peptides, it dies. But if the TCR is not able to recognize the self-peptides, whether on MHC class 1 or on MHC class 2. If it's not able to recognize the self-peptides, then it moves to the next stage. That means the TCR will not be able to attack self antigens or self cells. So the next stage, and that's the same thing that happens here. If for any reason the CD4 does not bind to the the MHC class 2, or the CD8 does not match with MHC class 1, this cell also dies by programmed cell death. So the same thing happens here. The cell also dies by apoptosis. And how does this occur? It is just the thymic cell releasing chemicals, majorly the FAS, to kill the cell. So it's a programmed cell death. Now, if the T cell receptor is not able to recognize the self antigen, it has passed. And we now call that negative selection. Okay, now moving to the next stage, we know that we have both CD4, right, and CD8, and CD8 molecule, CD8 molecule on the surface of our T cell, right? And we also have the T cell receptor. We want to know because at this level, there are two subpopulations of T cell. We have the T helper cells, the T helper cell, and the T cytotoxic cells. So some of the T cells will differentiate to form the T helper cells, and some of the T cells will differentiate to form the T cytotoxic cell, and it happens at this particular stage. So let's assume that another thymic cell comes along over here. Okay, if this CD4 is able to bind with the MHC class 2, if it's able to bind to the MHC class 2, right? Why downregulating this one, MHC class 1? That means that this particular cell will differentiate to form what we call the T helper cell, because we have downregulated the CD4 molecule. So we don't have a lot of CD4 on the surface, CD8 rather on the surface, but we have enough CD4. So we have more of the CD4 molecule on the surface of the cell. And any T cell that you have a lot of the CD4 molecule is regarded as the T helper cell. T helper cell. Now, what about the cytotoxic cell? For example, if we have more of the CD8 on the surface, right, binding to MHC class 1, that means that this particular cell is going to downregulate the CD4. So we're going to have very little or no CD4 on the surface of the cell, but more of the CD8 on the surface of the cell. And that will differentiate to form another type of T cell, which we call the T cytotoxic, because it binds more with MHC class 1. You understand? And it has more of the CD8 molecule on its surface. So you have CD8. Over here, you have CD4. So the CD4 cells are called T helper cells. The CD8 cells are called T cytotoxic cells. We also have another subpopulation of T cells, which are called the Tregs. The Tregs are T regulatory cells. What they do is they regulate the activity of the CD4 cells and the CD8 cells, that's the T helper and the T cytotoxic cells, by the time they've been pushed from the thymus out to the peripheral blood or to the bloodstream. So at this stage, the T cell has attained full maturity and differentiated to CD4 and CD8. So this is an overview of the development of T cell. What happens in the thymus?